So, well, my name is Valeria, as you said, Fernandez. So I work as a postdoc, it's like a part of the scientific staff of the stem cell core facility. This is a core facility that depends from the VIH, so the Berlin Institute of Health. And we work under Charité, so this is a big hospital in the medicine faculty in Berlin. So our main task is the reprogramming of somatic cells into iPSCs, and we do also all the downstream applications of the iPS. And we work in collaboration with different research groups, providing the technology, providing scientific support as well, and all the equipments that are needed to pursue, let's say, the research and the projects and topics. So the single cells, and in fact the isolation and expansion of clones is very important for different applications, mostly when it comes to related with the gene editing pipeline. So there we apply technologies as CRISPR-Cas, and we would like, for example, to generate knockouts or to make some base editing or some knock-in, for example. And in this case, we first apply the CRISPR-Cas in the bulk of the cells, of the iPSCs, and then it's very important to have a reliable and efficient method to isolate the cells, sort of to be sure that this is a clonal isolation, so that then after we can expand and we can generate a single cell line with the modification, for example, that we planned according to the gene editing. We have done this with other methods, like manual methods or after fax sorting, for example, and these cells are suffering a lot of being isolated, and also during the manipulation and the mechanical stress is quite an issue for them, and then the survival after seeding it was quite low. And with the cellenONE, what we have experienced, and it's very good for us, is that it's a gentle method, so we can do it in a fast way, and this is very good for the cells to come back to their comfortable situation in the media and in the plate that is the destination plate, let's say. And also we can keep part of the sample, so in this picoliter is so small the volume that we can also retrieve sample in case we need it. But for the sensitivity of the cells, this proved to be a good alternative, so we have kind of good survival for what we were used to before. Yes, so the most important steps, in fact, is for the survival of the cells and for the outgrowth of the clones, is to as well get the best conditions, so the good media for the cells, the good coating in the plates, and also fast manipulation for the media change. So it's all what is necessary for them to be comfortable and easy. And then another thing that is very important is the follow-up, so the monitoring of the growth of the clones, because along with an imaging, for example, device which is automated, we can have the pictures, and then after look into the colonies and already the shape of the colonies and how they look like will tell to us if the cell line is going well or if we need to repeat the process. I think, and it was quite nice also to share this with other research groups and also coming from the pharma. So the cellenONE, for example, is a nice, let's say, step in the process that is kind of quite automatic. And I think that for middle, let's say, size of labs or small labs is kind of an issue, this automation, because to connect everything together in a pipeline, that everything is working well and nothing is crashing in between, and not to have engineers all around all the time to solve the IT things. So I think that that's the thing to overcome, let's say, a little bit for the future. A lot of software improvisation, improving, so like this, different users can take it. But once this is working in the good direction, the interesting thing is that we can increase the amount of projects that we take, right? So that we can dedicate more time to the scientific part and not so much to the technical issues, and then run this in a higher throughput and get more projects are going and higher amount of results at the end.